From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 18th, 2023. Hello, Micronauts. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. And welcome to our Quality Quorum for today. It is the 8th episode of Matters Microbial. Thanks for listening and or watching. First, as usual, another denizen of my Wunderkammer. Most people are aware how much I love tardigrades because, after all, they've survived 5 global level extinctions. And I have a lot, as you've noticed, of tardigrade-related paraphernalia. Here's a beautiful needle-felted tardigrade. A picture is also being dropped into this, made by Frivolous Forest on Etsy. I will also post a link to the wonders of tardigrades in the show notes. For today's inspirational lapel pins, the first is a delightful depiction of an agarose DNA gel. I only wish it was fluorescent under UV light. Perhaps they're working on that now. Our second pin's in honor of approaching Halloween and our guest today. And trust me, the Pine Barrens is full of some great stories. Please see the links in the show notes. I love me some spooky things. So this second pin is the Loch Ness Monster urging us to believe in ourselves. I didn't have a Jersey Devil pin in my collection. The third week of my microbiology class has been #microbial fun, I must tell you. To develop skills with single colony isolations and the proper spreading of dilutions, we used bacteria-containing plasmids that have chromogenic pigment genes. As you can see in this image, kind of looks like Jackson Pollock gone crazy. And as you can see, the students had lovely plates to work with for this purpose. And in fact, one of my micronauts called them forbidden Skittles, something that I really enjoyed. In addition, we began our marine microbiological safari. Each group brought in a seawater sample from a different location and plated out dilutions on 2 different types of marine relevant microbiological media. It was interesting to see the different kinds of microbes that appeared even from that tiny droplet spread on a plate, as you can see in this general photo. I have a couple of goals when I do this, and one of them is to look for bacteria that actually consume agar. That's the solidifying agent in most microbiological media. And if you look at this where the red arrow is, you can see the example of such a colony creating a pit in the agar as it degrades the agar around the colony. And then, my favorite, the really weird-looking plates. And if you look at this particular plate, you see what look like polygon appearing colonies. This might be due to contact-dependent inhibition or perhaps antibiosis. My micronauts will find out. But as I say, it's always a joy. Now, ours is a microbial planet, from the top of the atmosphere to as far down as we care to drill where there's liquid water, and yes, perhaps on other worlds. And who better to tell us about some of the unusual places that one can find microbes than Dr. Lauren Seyler of Stockton University in New Jersey? Lauren's going to explain to us the relationship between the Pine Barrens, you know me, American history, legends, you know me, and geomicrobiology today. Welcome to our Quality Quorum, Lauren.
Thank you so much for having me. I'm so excited to have this chat.
It's been a long time coming, hasn't it?
It has. We've been mutuals on Twitter for so long, and yet we haven't actually ever sat down and had a conversation. So this is a great opportunity to do that.
So the first thing I'd like to do before you get into I was going to say the meat, but what would we say, the peptone of our discussion? Is to ask
Mm,
this question because a lot of folks are unaware of the differences between institutions that are grant PhDs and those that have primarily undergraduates. Could you talk a little bit about what you do at Stockton University?
absolutely. So, Stockton University was just recently granted university status a few years ago. It is a state funded school and it primarily serves undergraduates. We have a few master's programs and a few PhD programs, but not in my particular program. So I only work with undergraduates in the laboratory. So my research is a little bit more self-driven. I can't just pass off projects to a graduate student or a postdoc, like what would happen at a larger research university. But that means that I'm working more closely with students in the laboratory. I'm training them in a lot of basic laboratory skills. And in a lot of cases, I my lab is their first exposure to scientific research and how it's conducted, and it's really wonderful. On the flip side, my teaching responsibilities are greater than those of professors at big research institutions, because my job is a bit more teaching focused. So a lot of work gets done in the summertime. But I have students working with me all year round, and we have a great time. I've had so many students pass through my lab already. This is only my fourth year at Stockton, so I'm really excited and proud to get to work with students at this stage in their career when they're just figuring out what they like about science and how research really works. And so it's a wonderful opportunity for me.
There's nothing to match that look on their faces when they find something out that certainly they may not have known before, but perhaps something that no one has known before. And that feeling of ownership is everything, isn't it?
It really is. I think it makes all the difference, particularly for first gen students, for students from underrepresented backgrounds who may not really know as much about research, in particular research in science. It's just a completely different experience for them than what they would get in a laboratory course, for example, where everything's kind of planned out, and you sort of know what to expect from day to day. So they get the good of uh, discovering new things and generating new knowledge, but they also get some of the negatives where things don't work the way that we plan them to, or they're delayed because we're waiting for supplies to show up. And so they get the full spectrum of the research
We call
experience.
that the re and research.
Yes, exactly. Exactly. Didn't work. Try it again.
That's right, and we don't believe it till it happens 3 times, do we?
That's right.
You know
Replicate or lie.
it's really funny you'd bring this up because recently I had some students that were very, very uncomfortable with doing research, and I did this very simple idea where they would punch out using a biopsy device actually, a colony from a plate, resuspend, and then plate out to determine the number of CFUs, cells, per
Mm.
colony. And I did it specifically because I wanted them to do what they then did, which is say, is there a relationship between the size of the colony and the number of cells? Does it change from bacterium to bacterium? And I was so happy to hear that.
It's wonderful when we can generate those kinds of
Yeah.
experiences in a lab
It
course. And I think that too often those kinds of experiences aren't emphasized in a laboratory course. It's like, here's the formula, these are the results you're supposed to get. And if you don't get those results, you did it wrong. And that's
that's
it.
right. And that's why the cures, the course-based undergraduate research experiences are more and more the directions that most of us are going, which is great. Lauren,
Yeah.
I wanted to say I'm going to put a link up to your laboratory site, which your research lab site, which I adore. But I put this in the title, the thumbnail for your talk. By the way, my wife created that thumbnail. I hope you enjoy it the way I do.
Oh, I love it. I love
And you
it.
call your laboratory a deep lab, capital D, capital E, capital E, capital P.
That's right. So it stands for the Dark Ecosystems Energy and Production Lab. It's also a reference to Mystery Science Theater 3000. the Deep 13 lab because I love that
Well, there
show.
you go. So that's something that I hope folks will take a look at later on because there's a lot there, and so I'm gonna quit, yammering on a little bit, and I'm gonna turn things over to you to talk about your topic for today, which takes us to the Pine Barrens.
That's right. So, a lot of my research focuses on environments where light is unavailable. So, places like deep sediment, the aphotic ocean, deep rock. This setting is a little bit different. So, the Pine Barrens, first to just kind of give folks a general idea of what we mean when we talk about the Pine Barrens, right? Because I'm a New Jersey local, and so I grew up spending a lot of time in the Pine Barrens. But most people, when you talk about the Pine Barrens or New Jersey in general, people have very set ideas about what to expect if they were to visit New Jersey, and it's mostly based on the Jersey Shore and Newark Airport. So, um, the southern part of the state is, in fact, incredibly rural, and it's very spread out. And part of that reason is because the Pine Barrens are there. So the Pine Barrens are a very large, mostly unsettled area dominated by sandy, acidic soil, and as the name might suggest, pines, mostly pitch pines, lots of scrub oaks, some white cedar in the lower swampy areas. And it's a place that does not support most traditional agricultural crops. So it was very sparsely settled when white European colonists started coming here for those reasons. However, what we do have is a great water supply. And so the Pine Barrens is threaded with all of these beautiful streams and rivers and lakes that were great sources of transportation, not only to European settlers, but also to indigenous populations, the Lenni Lenape, who were here before the Europeans arrived. And also very, very pure groundwater. So most of the Pine Barrens exists on top of what's called the Kirkwood Cohansey Aquifer. And that water is acidic, it's corrosive, but it's some of the purest drinking water that's available in the United States. And so the Pine Barrens was set up as the very first national preserve in the 70s to protect that water supply. So even though we're situated in this um, corridor between these very densely populated areas, New York City, Philadelphia, Baltimore, DC, there's very, very little here in terms of human settlement. There's lots of open areas that have been preserved, forested areas. And as a kid, I grew up on the Jersey Shore in Ventnor, and I spent a lot of time camping in the Pine Barrens. And when I came back to this area and got the job at Stockton, what I really, really wanted to do was take my research into the Pine Barrens. There is very, very little information in the literature currently about the microbial ecology of this area, which is surprising because it's an incredibly unique ecosystem. It's the largest remaining example of coastal Pine Barrens forest left in North America. There's a little bit on Long Island and a little bit up in Cape Cod, but the piece down here in South Jersey is really like the largest remaining example. It's home to countless species of birds and mammals and fish and plants, some of which are endangered. But there's hardly any information about the microbial populations here. And so that brings me to the bog iron. So, as I said before, there's not much opportunity for agriculture in this area. We do have farms here, but the soil is not very good on its own. And there's only a few crops that will grow really well without heavily fertilizing and laying down, everything that's needed for agriculture. However, during the 18th century, iron was collected in this area. It forms where the groundwater, which is incredibly rich in reduced iron and anoxic, seeps upwards, comes in contact with the oxygen in the atmosphere, and then forms basically these very, very dirty iron oxide floccules. And I've shown some pictures. I put pictures in the Dropbox for the talk. It looks like this fluffy, velvety orange slime. It's quite beautiful, in my opinion. um, but these deposits over time would accumulate and combine with other organic material and form bog iron, which is a very dirty source of iron, but it's good for making things like cast iron pots and pans. There are ironwork fences and staircases still in the state capitol that are made from bog iron. And believe it or not, collecting bog iron was a really important source of iron for munitions for the Revolutionary War, particularly cannonballs. So very close to my house, about 15 minutes away, is a historic village called Batstow, and that was one of the primary furnaces where these munitions were made to support the revolution. And it was so important that the British actually had a plan to invade Batstow and burn it down, but they never quite made it there. So there was a nest of pirates, privateers at Chestnut Neck, which was attacked by the British and burned down. One of my ancestors was there at the time. His house was destroyed. And as the British were planning to move inland towards Batstow, they were stopped by the Polish general Pulaski, who was coming from the other direction. And they were like, oh, Pulaski's on his way. Okay, let's just abandon this plan. So fortunately, Batstow was never burned down, but it was considered to be a really important target of the revolution because it was supplying munitions for the war. So these mats of microbial growth eventually combine with other organic materials to form lumps of bog iron, which is mostly made up of very impure limonite mineral. And these lumps of iron could be collected and thrown into a blast furnace, so a very, very, very hot furnace. I believe that locally gathered clam shells and oyster shells were used to kind of fuel that blast furnace and to refine the iron and remove as much organics as possible. It was still a very dirty process, and you're never going to get very good iron out of it, but it's still good enough for cast iron and cannonballs. And bog iron continued to supply munitions to American troops for the War of 1812 as well. The industry was eventually abandoned because better sources of iron were found further west, which is why Pittsburgh and Detroit were steel towns, right? Because there's all these iron deposits that you could mine for and make steel from. Bog iron is terrible for steel. It's way too dirty. So what does all this have to do with microbes? Good question. I'm glad I asked it. So microbes facilitate this process of turning the reduced iron to oxidized iron. It's not something that happens abiotically where the iron just comes into contact with the air and oxidizes on its own. The microbes are actually accelerating this process. So there have been 2 whole papers about this particular process in the Pine Barrens. One dates from 1979 and the other one dates from, I think, 1986. Looking at the microbes that are responsible for this process. And they primarily looked at a particular type of iron oxidizing microbe, which is microaerophilic, meaning it likes a little bit of oxygen, but not too much. And it uses the electrons that it's pulling off the reduced iron in order to fix carbon. So, in my lab, we looked at these bog iron seeps, which are still plentiful throughout the Pine Barrens. They're everywhere. You just kind of stumble across them. And we were like, well, okay, so some folks a few decades ago found some microbes that are associated with iron oxidation, but they were using culturing techniques. They weren't using DNA sequencing. So we thought, well, probably a lot of diversity in these areas that was totally missed. And turns out we were correct. There are lots of iron oxidation strategies that microbes and microbial communities in these locations are using. It's not just about microaerophilic iron oxidation. There's also iron oxidation associated with nitrate reduction, iron oxidation that's fueling anoxygenic photosynthesis. And there are iron reducers as well. So geobacter is pretty plentiful in these spots. And we also see Shionella and some of the other classic iron reducers as
May
well.
I ask a quick question?
Absolutely.
I'm crying microbial tears of joy right now. And there's two reasons. One, on your website, you say something that's near and dear to my heart having to do with electrons. Do you recall what it is?
Yes, it's the Albert Szent-Györgyi, if I'm pronouncing his name correctly. He said that life is nothing but an electron looking for a place to rest.
Yeah.
I love
And so this idea that I introduced to students, why we breathe oxygen is a place to dump electrons. I have them hold their breath and raise their hands when it starts to hurt. And then I tell them they just have too many electrons. And that's what I do to first semester students to, kind of mess with their brains. But more importantly, microbes are so versatile in the sorts of electron acceptors and donors that they give. And this is a perfect example. I was also struck by what I can only call ferroautotrophy, the idea of working with respiring iron to power autotrophy. Now, is that not related to some theories of early Earth, which is a big interest of yours?
it. Is, in fact. And I ended up being really surprised the more I delved into these questions about these particular bog iron communities, how much the work there was related to other questions that I'm interested in part, the iron, because we know that iron is incredibly plentiful and was incredibly plentiful on early Earth and may fuel life on other worlds as well. In particular, in relation to autotrophy, but also these questions of how do microbes utilize these interfaces of redox, where you have highly reducing conditions on one side and highly oxidizing conditions on the other. So we have this very, very reduced groundwater coming into contact with the atmosphere, which is relatively loaded with oxygen. And you have microbes sitting right at that interface and using it to fuel a variety of metabolic strategies. And coming back to the electron quote, it's just electrons being shuffled back and forth between these two different redox states of iron. So you have the iron oxidizers on one side pulling the electrons off of the iron, and then you have the iron reducers putting that electron back on.
It's just
And
beautiful.
they may be, it is, it's beautiful. And another really fascinating thing about the water around here, or the water as we say locally, is we refer to it as cedar water. And the reason why is because the water here is this incredibly rich, dark brown, almost black color. It looks like really strong tea or coffee if you didn't add any milk to it. And it has this incredible irony, oaky smell. It's loaded with tannins from all the riparian vegetation along all of these waterways. And the tannins may actually assist in shuffling the iron back and forth between these redox states, keeping it in a soluble state. Because once you oxidize iron, it comes out of solution at near neutral pH. And the water here is on the acidic side. It can go all the way down to pH like 4.5, pretty acidic. But the iron is still going to want to come out of solution and form these floccules of rust. If these microbes are using these tannins as a way to shuttle the iron back and forth, I mean, that's just really interesting. And that's something that we haven't been able to delve into just yet, but it's on our to-do list. So it's just an all around fascinating
Do
place.
you know, I have what I call the deadly centrisms that we all deal with, and one of them is oxycentrism. And this is a
Mm.
really
Oh,
good example. The whole idea of the different redox states of metals, one being less soluble than the other, so that in the process of, quote, breathing, something that was soluble becomes insoluble is really common, but we don't see it because of us using oxygen the way that we do.
yes. And I think getting that idea out of students' heads that life requires oxygen is its own battle,
It is.
right? Because they're so used to thinking that, oh, everything that's alive needs oxygen to breathe, and that couldn't be further from the truth. And I love thinking about, what about life without oxygen? What about life without light? The kinds of things that we take for granted as just being required for life to work,
No,
right?
100%. I mean, we are fellow travelers, my friend.
I love that. I do.
So please go on. I mean, I don't wanna direct you too much because this is so exciting, and I'm just afraid I'm gonna say too much.
Well, what we're finding right now is a lot of diversity. Not just in terms of the actual species of microbes that we're seeing, but in terms of the different types of iron cycling strategies that are available to them.
Wow.
I'm just delving now into a bunch of metagenomic data that we got last summer. And we've generated bins, and I'm looking at some of those bins and trying to figure out, all right, who is doing what? And we're seeing a lot of the microbes that were identified in these early papers, the ones that are culturable and very well classified. But we're also seeing iron cycling strategies in species that are not traditionally associated with those strategies, which would make sense. There's so much iron being moved back and forth in these environments that it stands to reason that species that maybe we don't think of as iron oxidizers or iron reducers are in fact oxidizing and reducing iron because it's there. It's there as a source of electrons or a sink of electrons. Um, so I'm still really delving into that research. This past summer, we went out again and we did some more sampling and we extracted for DNA, but also RNA. So now not only can we look at the genes that are available to this community, but also which genes are actively turned on, which makes a lot of difference because just because a gene is there doesn't necessarily mean that it's being utilized. So we have plans to look a little bit more at what genes are actively being used. Are there seasonal cues with these seeps? Because the previous literature suggests that these bog iron seeps kind of pop up in the spring when the growing season starts and then they kind of die off with the end of the growing season. However, with climate change, we've had some really, really mild winters, and I've seen seeps with active or active looking blooms in the deep winter because the weather is so
Wow.
mild. So it's a possibility that these are still maybe not as active, but still active in the wintertime because we're not getting as much below freezing temperatures as we used to in the past.
Y
So, but anyway,
I was gonna say, I recently did an episode of This Week in Microbiology, and I don't know if you saw the paper, but there's some evidence that continental drift began because of iron metabolism on early Earth, with
really,
magnetite being dense enough to lead to that original fracturing. And this is from this is from a geology paper. I'll send you a link. It's pretty interesting.
yeah, I'd like to see
Yeah, it was
that.
fun.
Um, it's really interesting to think about because iron is it's one of those things that it's a micronutrient, right? So as microbiologists, we're usually thinking about the big ones like carbon, nitrogen, sulfur, phosphorus, the things that make biomolecules, oxygen. Iron is one of those things that almost every living thing requires, but we know so much
Yes.
less about how it's cycled and how it's made available. And we've even found in these communities a magnetotactic microbe, which I
Oh.
love.
Oh,
They're so
what's
cool.
the morphology on that one?
No idea. We're just looking at the metagenomic data at this point, and we've found the genes for
Oh, I see.
producing the magnetosomes. Um, and off the top of my head, I can't remember what species it was
It's
associated with.
okay.
It was just the one. But it was so cool. It's like, oh man, for those in the audience who don't know, there are microbes that make an internal compass and move along the magnetic field of the Earth for some reason. There are theories about it, but we don't know exactly why they do this or why it's an
It's
advantage.
because they're awesome, Lauren. That's the reason.
Yes, exactly. It's for the clout.
Well, you know perfectly well, with all due respect to Beyonce, who run the world, and it's microbes. There
Micro,
you go.
absolutely, absolutely.
So tell me a little bit about your
So,
work with students. I mean, can you give us a wonderful success story?
oh my gosh, I have so many great students. Um, I've had students who are going on to pharmacy school. I have a student who's currently working with the NIH. He's been doing a fellowship with the NIH, and now he's looking at graduate school. Um, Students who went on to be nurses. I mean, I have a student who was going to be a vet, and now she's like, I don't know. I think I really want to do scientific research with microbes. And I was like, right on, you know, not to disparage the medical
No.
field, but it always warms my heart when people are like, you know what, I think research is really for me. So I've just had so many wonderful experiences taking students into the field. And this past summer, I only had 3 students, which is a small cohort for me, but they so quickly picked up how to take samples, how to take geochemical measurements, where to sample from, how to store things. I train them in DNA extraction, library prep for sequencing. I mean, pretty much the beginning to the end as far as all of the field and laboratory work. And now, we're getting ready to look at that data and hopefully get a nice paper out
Oh,
of it.
that's wonderful.
Um, it's just fantastic. It's so great to involve students in that work. Um, we've done a little bit of culturing of these microbes from the bog iron with some success. Um, we're still playing around with the conditions. There's numerous enrichment strategies, and it's a little tricky because a lot of these iron oxidizers like a little bit of oxygen, but not atmospheric levels. And so you have to set up culturing conditions where oxygen just sort of percolates down into the
Right.
medium and the microbes settle right where that concentration of
Stratify,
oxygen
yeah.
is.
Have
Yeah, it's a Goldilocks situation for sure.
are you familiar with Slava Epstein and Kim Lewis's work at Northeastern? Where they
No,
make and
no, I'm not.
I'm not going to describe it very well. Again, if you remind me, I'll send you a link. They kinda make like a miniature aquarium so that they can take something they're trying to isolate and put it in chemical contact with the environment that it's in, because there are many microbes that can only grow in the presence of other types of microbes, which is, I think,
Right.
probably how most microbes actually live. Um,
Yeah.
coli centricity, by the way, is another deadly centrism. It just makes me wonder if you were able to dump, or dump one of these particular devices in there, you might be able to have something growing in isolation in there, but in contact with the outside seep.
Yes, I think that would be extremely helpful because there are certain species that we know we can grow in the lab. And I know if I go to these sites and I take a sample and I enrich for them, I'll be able to
Yeah,
grow them. And they're pretty well characterized
yeah,
already. There's so much more diversity in
yeah.
there. And like you say, I think there's a reason why we've only been able to culture 1% maybe of what's actually out there in the environment. And that's just because most things don't want to grow in
That's
isolation.
right. Isn't that crazy?
It is. It's
I
wonderful.
find siderophores all the time in isolations, and what they'll do is they'll sometimes steal iron from the media so things can't grow around them, or in some cases, donate it to other things that can't grow without them. So it's just
Right.
wonderful.
Oh, yeah. I think that when you think about microbes as a community rather than as individuals, you start to really get at the complexity of microbes because when I was an undergrad, I took a general microbiology course, and they're like, here's the microbes, look at them under the slide. And it's like, that's a couple of dots. Okay, cool, whatever, and I didn't really think that they were interesting. And I think that's most students' experience of
Yes.
bacteria, right? Is like, oh, it's some specks under a microscope, whoop de doo, whatever. But when you think of them as these incredibly complex communities with communication between all these different species and sharing and competition and cooperation, they're really like these very complex city
They are.
states where you have all of these like incredibly complex interactions going on. And then when you put it in the context of the environment and you think about the interaction between life and non-life, right? Rocks, water, air. These are the things that really like keep me awake at night in a good
So
way.
you're aware of my hashtags OMG for overwhelming microbial greatness, which clearly you're into.
Yes.
Also, I
Oh yeah.
have WTM for what the microbe? Because you just don't wanna believe it's as cool as it is.
Oh, it's amazing. We're constantly learning new things about these deceptively simple organisms,
Well,
right? Things that like theoretically we thought could happen in terms of like different types of metabolism. But then one day we observe it somewhere, like a wastewater treatment plant or something, and we go, "Oh, it actually can happen."
like I say, it's first evolved, last extinct. And I've been saying that for decades, and it's absolutely true, and we tend to forget all that. So they are the small
Yeah.
masters to me, right?
Oh, absolutely. The first in, last out, the true heroes.
Well, and that's why we're microbial centrists here. That's what it comes down to.
Yeah,
Do you
absolutely.
I could go on for hours, but I obviously, you have things to do. I have to teach this afternoon. So why don't we talk a little bit about well, you know what? Let me back it around. Is there anything special that you wanted to say to wrap up your research to the listeners and viewers?
Um, I think that the most important takeaway from my research in particular is how there are these fascinating relationships and interactions between life and the non living world, right? The environment around them. And they're happening literally everywhere. I mean, these places where I go to sample are mostly historic sites where bog iron was collected and refined and found formed this booming industry for about 150 years and then died out. And we go there now in the Pine Barrens, and there's remnants of these old buildings, and some of them have been preserved as historic sites. Some of them are just abandoned in the middle of nowhere, but they're minutes away from my house. I don't have to go all the way to the bottom of the ocean or all the way to Yellowstone or all the way to the semi-ophiolite in Oman, all places that I've been, right? And they're all fascinating in their own right. But I don't have to go that far from my own front door to find all of these incredible interactions. They're literally everywhere. We are surrounded by an invisible world that is incredibly complex and has so much of an effect on everything that we are as living beings. And so that's I think at the heart of this microbe centricity, right? Is that microbes are really running the
Oh
show. They
yeah.
just don't get the credit.
It's a microbial planet. I couldn't agree more. And the interactions are beautiful besides. And you see, I was good.
Yes.
I didn't even bring up spooky stories, though Halloween on some of those ruins would be awesome. Don't you think, reading stories by
Oh,
candlelight?
they're absolutely 100% haunted. And as I mentioned, my ancestors are from this area on my dad's side. And I have, I can go to a local graveyard and go, yeah, related to them, related to them, related to them. Like I have a branch of my family that just settled here when New Jersey was still a very young colony, and they just never left. They were here for
The
hundreds of
Dark
years,
family is with us at all times, Lauren.
all times. And uh, I know we promised that we wouldn't go off on a very long tangent about this, but I am in fact related to the Jersey Devil. So I had to bring that up for the
So
club.
no, absolutely,
But
you own it. But the thing I would remind you is I did put a link to some of the most important stories having to do with the Barrens, and I hope people can read about that and give you proper credit for being like a descendant of the Jersey Devil.
that's right. Yeah. So there's some really fascinating local history around here, and I think a lot of it goes overlooked, and it's wonderful to get to weave my research into the history of this place as well.
Well, it has been an honor and a pleasure chatting with you today, and I've gotten a lot out of it. If there are things that I'm supposed to send you because we've talked about them, please let me know. And
I'll remind
and you should know that everybody who puts up with doing a segment with me gets a little prize. You'll be getting that soon, just so you know. I
you.
know.
It's been a pleasure, Mark. Thank you so much for
Oh, it was a
having
lot of
me.
fun. It was a lot of fun, and I wanna wish, you and your family and your students the very best.
Thank you. The same to
Thank
you.
you. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes, things you must read, for today's episode can be found at microbe.tv/mm. If you like our work, consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Lauren Seyler, who is a pleasure to talk with, you can find in the biology department at Stockton University in New Jersey. Many thanks to David Renata for superb editing and Reber Clark for wonderfully quirky music. I hope you've enjoyed being part of our quality quorum for today. See you next week on Matters Microbial.